and good morning, good afternoon, and good evening. Welcome to the ICAP Grand Rounds webinar. We're going to get started in just another minute or two as we give folks a chance to join. So please stay with us. Okay, good morning, good afternoon, good evening, and welcome everyone to today's ICAP Grand Rounds. I'm Dr. Jessica Justman, the Senior Technical Director for ICAP at Columbia. And today we're going to hear about insights from successful virus outbreak responses. I think as we described in publicizing this session, Marburg virus disease is an often fatal hemorrhagic fever. It's clinically similar to the Ebola virus disease. And this past fall in 2024, Rwanda experienced one of the largest recorded Marburg virus disease outbreaks. And the majority of these cases were among healthcare workers. In response to this outbreak, Rwanda's Ministry of Health launched a comprehensive national program that included widespread public health awareness promotion and a number of other steps. And this also included the distribution of investigational vaccines and therapeutics. We're going to be hearing about this response in more detail from an expert from the Rwanda Biomedical Center. And we will also hear more about the development of the Marburg virus disease vaccine from an expert from IAVI. So let's go ahead and introduce, before I introduce our first speaker, we have a French translation available this morning. So if you'd like to hear the French translation of this webinar, please turn on the French channel by clicking on the interpretation icon at the bottom of your screen. It looks like that circle. If you don't see that on the bottom of your screen, click on the circle that says more, and then you will see the word interpretation. We have an interpreter who is interpreting my instructions. So I hope you are able to find your way to the French translation. After the presentation, we will have about 10 or 15 minutes at the end for questions and answers. For questions for our panelists, please use the Q&A box that's at the bottom of your screen. Okay. So I'm very pleased this morning to introduce our first speaker, Dr. Edson Rwagasore, who is head of the Public Health Surveillance and Emergency Response at the Rwanda Biomedical Center. Dr. Rwagasore is a public health physician and epidemiologist, and he leads Rwanda's national surveillance systems, outbreak investigations, and emergency health responses to protect public health. He's also the program director for the Rwanda Field Epidemiology Training Program, FETP. where he's building the capacity of the country's next generation of epidemiologists. And he played a key role as the lead for surveillance during Rwanda's successful response to the 2024 Marburg virus disease outbreak. Dr. Rawagasore, please go ahead and give us your thoughts on the Marburg outbreak. Thank you.
Yeah, thank you so much, Jessica. So I would like to share some of the lessons learned during the Marburg response in Rwanda. So our next slide. So it was until 27th of September, 2024, where Rwanda, we felt much more safer. And despite being within the region, which is becoming more and more prone to viral hemorrhagic fevers, it was until this day that where we recorded our first case of Marburg. It's a deadly disease. And when this outbreak occurred, a lot of questions was raised, but also I thought about mainly three things. Mainly, one, first of all, this disease, as a lot of people think about what was to be the extent of this outbreak and what would be the source of infection. But on my side, also thinking about this actually came during the wrong timing when we are struggling with the mpox, trying to contain the spread of mpox within 60 days. And at the same time, actually, it also affected. We'll think about how it's now affecting our healthcare workers who were high
I think we're having some trouble with the audio.
population movement. So containing this kind of fear, but at the same time strengthen on how we can move even faster to be able to contain it. Oh.
Dr. Edson, can you hear me? I think we're having some trouble with your audio.
So sorry. See the slide moving?
No.
We are hearing you, but the slides are not moving.
Yes. All right. Yes. So the second slide is on. Actually, this outbreak was not the first outbreak. This was the 13th outbreak that was recorded since 1975. And it's caused by the virus. Marburg is caused by virus. And actually, the first case was recorded in German Marburg, and that's how the name actually came out. So you can see most of the African countries are most affected, including the DRC, including Uganda, Kenya, and Angola. Next slide. So the transmission dynamics around Marburg, mainly this starts with the spillovers from bats, and you can see that the virus maintains itself within bats known as food bats called Rosetta's Egyptiacus, and over time, actually, most of these bats live and do roosting within caves and mines, especially at night, and actually live at night for foraging. And most of the time, the spillover can occur to primates, but also it can occur also to human due to the fact if there's any kind of interface between human and animals. Then human-to-human transmission, which is actually occurs very faster. it's also very possible and actually affects most of the healthcare workers, as it occurred in Rwanda. Majority of healthcare workers in most of the outbreaks are mainly being affected. And then over time, we also see persistence of Marburg virus within body fluids or among MVD survivors. Next. So it has different phases. So many people, whenever they think about Marburg, they think that it's a deadly disease, but also most of the time they think that it's a viral, it's a viral magic fever, and this should be appeared or should be presented as people who present with bleeding. But majority of cases come with just malaise, high fever, chills, severe headache, and myalgia. And on the phase 2, where actually there's start of early organ phase, they also observe kind of neurological symptoms and also impairment of some of the multiple organ functions. So that's why most of the time these kind of outbreaks are missed, and they are recorded or they are identified when there's actually larger outbreak where majority of people have been affected. Next. So story started in Rwanda, where some of the healthcare workers actually, I had a call with them, and they told us that there's they're feeling unwell, and we identified actually clusters of cases within the two major referral hospitals. One is King Faisal and Sashka, which are main referral hospitals, which actually have the biggest ICU in the country. So also, during this outbreak, after identification of cases, rumors kept on circulating, where we had some of the media coming in, jumping in, just to push that there might be something wrong. But at that time, we already had started to identify cases, and then we had to communicate this as soon as possible. Next. So the first step, after just identification of the first cases, we had kind of a 72 hours clock running. And during this period, it's a timing clock that we use whenever there's such kind of a bigger outbreak. And it's quite important, since every minute of that clock counts. And during this outbreak, our first day, we actually focused on how can we make sure that we are well coordinated, how can we make sure that we do communicate in a proper way, and how can we make sure that people are isolated. But the most important, how can we make sure that countries are also, within the neighboring countries, are also prevented. So we had put all kind of interventions, including limiting people movement from out of the country, especially contacts and cases, and isolation of cases. And the most key aspect is how we engage different partners. You can see on those photos, our minister was engaging with different partners, WHO, the US ambassador, and all other embassies and stakeholders were onboarded, that with the proper communication, we're able to at least communicate to everyone and to understand what's going on, and also avoiding any kind of rumor. On the other side, you can see the structure. It's a very known structure of our command post, where we had to put in place different pillars to make sure that everyone is actively engaged, from the AP, logistics, but also communication. Next. So over time, with the great work of contact tracers, and by making sure that every case has been isolated, all contacts were put into kind of a bubble to limit any kind of transmission beyond that bubble. So since the good news, since most of the cases that were affected were from the healthcare workers, and we are able, at the initial phase, we're able to at least put everyone in such kind of a bubble within a specific hotel, where we are able to limit any spillover or transmission at the household level. So we had a good signal on the third week, as you can see, where we are able to at least cut by half the number of cases, and then later we had to continuously contain the spread. So this was a kind of a good graph for us that showed that there's a good yield in terms of what kind of interventions has been put in place. So you can see the most affected were healthcare workers, other students working within those kind of settings, and also we had other patients that actually were got the infection from the hospital itself. Next. So the most important part is where the team of contact tracers were able to identify the link and also provide the insight of the index case. And we were able to, after further investigation and looking at the medical files within one of the ICU, we were able to identify the index case and link that case to one of the mining sites. This was one of the known mining site within the city of Kigali, where after further investigation, actually the team did investigation and identified that there are actual food baths within this mining site. It's a known site within Kigali, but actually we never thought that this site has food baths. And then after further tests, we were able to reveal that there are actually additional people who already been exposed. Actually, 14 people had already been exposed with Marburg virus, but they didn't really show the symptoms. Next. So these are our heroes. Our heroes were frontliners, people who spent much of their time on bedside, treating patients, and spending a lot of time at risk. So you can see one of the doctors here on the right side who spent almost 8 hours within PPEs. But also we had to make sure that we prevent anyone to be infected with proper IPC measures that was put in place. Actually, only one doctor was infected after the transmission within the health care facility. But we didn't want even this case to be recorded, but there was a kind of a gap in terms of IPC. We already had one patient that was linked from the hospital level. Next. So another key intervention that actually changed the paradigm in terms of how we're intervening is the ring vaccination. Just within 9 days, we're able to acquire vaccines, and also we're able to at least vaccinate the most at high risk healthcare workers working within these kind of settings, but also the most important are the close contacts. So whenever we identify a case, we had to vaccinate everyone who is very close to that patient. Next. So all this yielded to good results where we vetted how people view Marburg, which is actually deadly, but actually we were able to keep the number of mortality below 24, where we had the case fatality rate at 22.7. Next. So since the healthcare workers were most affected, we also had kind of both psychosocial impact on our health systems. And, these are people who are working in ICU, the main big ICUs within the country. So we are working closely with the team from Rwanda Medical Association to make sure that we do support them on psychosocial support, and also kind of continuous follow-up to support them as as post-MVD survivors. Next. So we also had kind of evidence just to document. These are just lessons learned for Rwanda, but also can be applied for other countries. And we are trying to document every single step that made us a success on containing the spread, including the genomic and transmission dynamics of this outbreak, linking this outbreak to the index case and to the mining site, which is very important as we be moving forward to prevent future spillovers. Next. So the key next steps, as you can see, we have established 4 strategies. One is on risk-based geographic mapping of the risk, where we have mapped all high-risk areas, including mining sites, where these food baths are known to be roasting, but also looking at species distribution, but also the intermediate hosts that can also be the risk for transmission. But also, at the same time, you are working closely with the other colleagues, having GPS being tagging on to see the pattern, movement pattern, but also the seasonality in terms of shedding of the virus from bats to human. Next. So this is just how we're doing with a quite good sampling strategy. As we move forward, we'll be improving on how we do sampling, but most important, we're also learning from the bets from colleagues around the world globally to see how we could prevent the future outbreak in Rwanda. Next. So this is just the plan for the future plan for the next key intervention, mainly on bats and GPS. Next. So thank you so much. Maybe I just want to echo on this. We couldn't have done this work without partnership, and it's just to thank every partner who, especially ICAP, who actually onboarded to support during the response. So this kind of outbreak are always lessons for the future preparedness, and we're looking forward to even work better, to improve better for the new epidemics or pandemics. Thank you so much.
That was great. Thank you so much, Dr. Edson. That was really terrific overview and very clear. Our next speaker is going to talk to us about vaccine development for Marburg and other viral hemorrhagic fever diseases. Dr. Mark Feinberg is president and CEO of IAVI, where he leads a global team working to advance the development of vaccines and other biomedical innovations to protect against HIV, tuberculosis, emerging viruses, and other public health threats that disproportionately impact people living in low-income countries. Prior to joining IAVI, now about 10 years ago, Dr. Feinberg served as Chief Public Health and Science Officer with Merck Vaccines, and he led a range of research initiatives to address unmet health needs in low-income countries, including the coordination of a private-public partnership to expedite the development of an efficacious Ebola Zaire vaccine. So I think, Dr. Feinberg, please go ahead and tell us about the vaccine development story. Thank you.
Thanks very much, Jessica. I just want to offer my congratulations and amazement to Edson and his colleagues in Rwanda for their very effective response to a novel Marburg outbreak. I'm going to focus my attention on the development of Marburg vaccines and to give you an idea about where we are in that process. Next slide. Just by way of background, for those of you who may not be familiar with IAVI, we're in essence a not-for-profit biotechnology company. We have capabilities from discovery, translation, clinical evaluation, and extensive community engagement, with a focus on developing much-needed vaccines and monoclonal antibody products addressing diseases that disproportionately impact people living in low-income country, including HIV, tuberculosis, a range of emerging infectious diseases, as well as select neglected diseases. Next slide, please. To basically, show you our portfolio, and you can click through the animation here. We focus on vaccines against a range of infections that I just mentioned, but in the emerging infectious disease space, our lead candidates include a Lassa fever vaccine, a Marburg vaccine, an Ebola Sudan vaccine, and a Crimean Congo hemorrhagic fever vaccine. All of these are emerging infections for which there are currently no prevention modalities available for them, but we're optimistic that these can be developed and are very dedicated to developing them. Next slide, please. To give you a sense about our emerging infectious disease portfolio, you can go to the next slide. And why we're working on this, I mean, I think all of you know that emerging infectious diseases like Ebola Zaire, Ebola Sudan, Marburg, et cetera, I mean, it's not just your impression that these diseases are emerging more frequently as well as in unexpected places, because that is, in fact, the case. And it just really reinforces our need to not only have the modalities such as vaccines and treatments that can be addressing these, terrible diseases, but also the systems and partnerships and processes in place like those that Edson referred to rapidly and effectively respond to newly emerging outbreaks, because they're here and they're going to keep emerging because of, whether it's climate change, migration patterns, war disruption, population growth, urbanization. All of these are contributing, we believe, to an increased risk of emergence of a range of zoonotic infectious diseases emerging from reservoir animal hosts. Next slide, please. And this isn't, again, just hypothetical. If you actually look at where recent Marburg outbreaks have taken place, Edson alluded to this a little bit, but to get a bit more specific, if you look at the places where recent outbreaks have emerged, including in Guinea, Ghana, Equatorial Guinea, Tanzania, and Rwanda, these have all occurred within the past 5 years. And for each of these, it's the first outbreak of Marburg in those countries. There was another outbreak in Tanzania, but Tanzania had seen previous outbreaks, but it is spreading more widely, more frequently, including locations where the disease has not been previously seen, which is obviously important to take into account. Next slide, please. The vaccine that was used in Rwanda is not the vaccine that IAVI is developing, but I did want to highlight it for you. It's a vaccine, so-called chimpanzee adenovirus vector. The same kind of vector system that was used in the AstraZeneca COVID vaccine is being developed by the Sabin Vaccine Institute, and they had partnered with the Rwandan authorities to include their vaccine in the ring vaccination trial that Edson referred to. Next slide, please. The vaccine that IAVI is developing is based on the vesicular stomatitis virus-based platform, which is a platform which is the identical platform and using the identical approach that was used in the successful Ebola Zaire vaccine development effort that was undertaken by Merck, along with a range of global partners, and that was demonstrated to be close to 100% efficacious in a ring vaccination trial that was done in Guinea towards the end of the 2014 to 2016 Ebola Zaire outbreak. So it is a licensed platform which has demonstrated high-level efficacy with a single-dose vaccine with a rapid onset of immunity. And what's interesting is those properties seem to be shared across a range of other vaccine candidates that are based on the same vesicular stomatitis virus-based platform. And the way this works is that you substitute the natural glycoprotein for the vesicular stomatitis virus, or VSV, with the glycoprotein from the pathogen you're interested in vaccinating against, whether it's Ebola Zaire or Marburg, so that this virus now displays on its surface the glycoprotein from that heterologous pathogen, and it displays that to the immune system in a way that is essentially identical to what the natural pathogen does. But because it doesn't cause disease, it can induce protective immunity. And you can see the differentiating features of this platform on the right-hand side. And importantly, in addition to being highly efficacious with rapid onset of immunity with a single dose, we're able to use a common manufacturing platform, which means that if you're able to make one of these vaccine candidates, you should be able to manufacture all of them, which is especially important for vaccines for which the demand is unpredictable and it may need to be scaled up and scaled down. Traditional manufacturing platforms don't work for that, but if you could make a portfolio of candidates that you could cycle through over time, you'd have a much better public health response. And that's the goal we've been pursuing from the beginning of this effort with the portfolio of candidates you see here. Next slide. Just to show you where these are, our Lassa vaccine is undergoing phase 2 evaluation in West Africa. Our Ebola Sudan vaccine was in phase 1 studies in the United States, but it was used in the Uganda ring vaccination trial that just took place in the past couple months to deal with their most current outbreak. Our Marburg vaccine candidate will be entering phase 1 trials later this year, and our Crimean Congo hemorrhagic fever virus is still in preclinical studies. Next slide, please. Just to give you a bit more background about the vaccine we're developing and the promise of it. Traditionally, the gold standard animal model for viral hemorrhagic fever diseases is non-human primate challenges, and this is a very stringent model where animals are infected with highly pathogenic viruses intramuscularly, and it's very, difficult to protect against that, as you can imagine, with such, virulent viruses. So when you see protection in this model, it is actually quite meaningful. And the way the study is done is the animals are vaccinated on day 0 and then challenged with the pathogenic virus on day 28. And what you can see on the left-hand panel is across a range of doses from 2 times 10 to the second all the way up to 2 times 10 to the seventh, there's 100% protection with a single dose, which is very impressive. If you look at the right-hand panel, which is showing viremia in the animals, it's even more impressive to see that you don't actually see detectable viremia in any of the vaccinated animals, whereas the control animals all become very viremic and progress to a fatal disease. So this is very similar to what was seen with the Ebola Zaire vaccine that is now licensed as Ervebo. And this, goes to my earlier point about it's impressive how what you see with one VSV vector against a viral hemorrhagic fever seems to be recapitulated with others. Next slide, please. The vaccine does induce both binding antibodies and neutralizing antibodies, which provide you with a valuable marker to compare how the studies in the non-human primates compare to what you see in people, and will be important inputs into the regulatory package that's put together to support licensure of these vaccines in the future. Next slide, please. We are planning, as I mentioned, to conduct a phase 1 study later this year, obviously with a focus on primarily a safety endpoint, but also looking at immunogenicity as well. And we're excited about that. Next slide, please. By way of analogy, I wanted to also give you some background on a recent outbreak of Ebola Sudan, which is related to the Ebola Zaire virus, but the Ebola Zaire virus vaccine does not protect against Ebola Sudan, so we need another vaccine against this pathogen as well. And you can see the number of outbreaks that have taken place across West and Central Africa, and you can see a number of outbreaks taking place in Uganda. The most recent one was in 2025, earlier this year, beginning in January. So this is another disease where a vaccine is greatly needed, not only to protect healthcare workers, but to respond to outbreaks and to be able to contain them from the very earliest times that they emerge. Next slide, please. We had partnered with investigators in Uganda and the Ministry of Health in Uganda when they had a recent outbreak in 2022, and partnered with Merck to expedite, the sort of finalization of the manufacturing process and shipping the vaccine to Uganda as soon as we could during that outbreak. That process at the time took 6 weeks, which was really a record-breaking period of time at that point in time, but it was not adequate to address the outbreak. Unfortunately, the public health authorities in Uganda were able to bring that outbreak under control actually before the ring vaccination trial that was planned could actually start. But it was fortuitous that we had the vaccine in Uganda, as you'll see. Next slide, please. This is the results of the phase 1 study that was done of this vaccine. Again, it's endpoints looking at safety and tolerability, as well as immunogenicity across a range of doses. And on the next slide, you can see the results of this study, which are very similar data in terms of both binding and neutralizing antibodies against the Ebola Sudan virus. Again, this is very similar to the kinds of results that we've seen with other VSV vectors against Ebola Zaire and Marburg. Next slide, please. As I mentioned, it was fortuitous that we had shipped the vaccine to Uganda in 2022 because when there was an outbreak that arose in January of 2025, it was possible to actually initiate a ring vaccination trial. And kudos to the healthcare workers and the public health authorities in Uganda for doing this, and the WHO who helped contribute to this. They were able to initiate a phase 3 ring vaccination trial within 4 days of the actual first report of an index case of Ebola Sudan in Uganda, which really demonstrates that having a public health response that is enabled by having stockpiles of the vaccine locally situated and a really sort of vision for how to implement a public health response as quickly as possible is of profound importance. So it's not just having the vaccine, it's having it at the right place and being able to deploy it as quickly as possible. Fortunately, the outbreak in Uganda is now under control. We don't believe that there will be enough cases to actually evaluate the efficacy of the vaccine, but I think it was a very successful response, and I congratulate the teams in Uganda. Next slide, please. This is basically the ring vaccination design that was used. It was different than the one that was used in Rwanda, but very similar. And if you click the animation to the one that was done in Guinea for the Ebola Zaire vaccine. So this is where you have 2 rings. You don't randomize individuals, you randomize the rings to either immediate vaccination or delayed vaccination and compare the infection rate in the different groups. And that's what was done in Guinea in 2016. And that was what was done in Uganda most recently. Next slide, please. When you're thinking about bringing these vaccines to licensure, you can't, because they're sporadic in their emergence and often relatively small in terms of magnitude, doing traditional vaccine placebo-controlled trials demonstrating efficacy is not necessarily practical here. So there are opportunities for animal rule licensure pathways that the FDA has, as well as, because we are working with a platform that's already the basis of a licensed vaccine, our discussions with regulatory authorities suggest that we might have a more expedited path of using a clinical immunobridging approach where we show essentially similar immune responses between the different VSV vectors, whether it's Ebola Zaire and Ebola Sudan and Marburg. Next slide, please. Just to close with a couple of comments about the future, obviously these vaccines are only going to be useful if they're shown to be safe and efficacious, and they can be provided to people who need them in an affordable and sustainable way. And in that regard, we're very happy to have established a partnership with the Institut Pasteur in Dakar, Senegal, which is one of the only vaccine manufacturers in Africa and the only one that makes drug substance manufacturing, in their facilities. And, we have transferred the rights to manufacture all of our VSV-based vaccines against Lassa, Marburg, Ebola Sudan to IPD. And we're really excited to have a partnership with them to enable them to manufacture them and supply them to the African continent and beyond. Next slide, please. Obviously, as Edson said, all of this takes a partnership, especially when you're talking about meeting the needs of people living in low-income countries. I know the ICAP team knows that well. And I just want to acknowledge all the great organizations that we've worked with for our end-to-end development pathways. Next slide, please. I just wanted to close with, next slide, with a thought. This is a photograph of a young girl from Liberia whose community had been heavily impacted by the Ebola Zaire outbreak in 2014 to 2016. And when she heard the news of the successful efficacy trial in Guinea. This was a photograph taking of her message to the broader community of people who worked on that. And I think in an era where science is being placed in a very bad light and really sort of disabled from doing all the good it can do in the world, I thought this was an important message to share with you. So thanks very much for your attention, and I'm happy to address any questions you might have.
Okay, well, thank you so much. That was really great. And we do have a number of questions that have come in, and I have a question or two myself. Some of the questions have been addressed, but I still think that they might be interesting to hear about in a little more detail. There was a question to Dr. Edson about whether the 717 strategies were implemented and what were the challenges encountered during implementation. So I thought perhaps, Edson, maybe you can respond to that verbally and remind people what that 717 strategy is, and talk about what the experience was with the outbreak last fall.
All right, thank you so much, Jessica. So for the 7-7, it's a global goal where we are supposed to identify cases within 7 days, notify within 1 day, and then at least respond within 7 days. So this we try to align on some of the data that we have during our after action review that actually occurred after the containment of this outbreak. And we found out that we are doing good in terms of the one, that's notification, and also the response, but we lag behind in terms of identification of cases. But this is very common for most of the viral match fevers, since the outbreak they don't show typical symptoms for, and the symptoms actually tend to occur or present when it's kind of late stage, where there's several cases that has occurred. So we kind of lag behind, and actually we went even beyond the 7 days. But with this, we had kind of lesson learned, and we're trying to improve on our systems. And as we move forward, we had put in place what's called kind of a triage for everyone coming in, checking the risk. And this was the first outbreak in the country. We never expected to have a Marburg. So when we had this kind of cases, the initial thought were these cases being imported, since we had never been recording such kind of outbreaks. So I think these are just some of the lessons, and we keep on improving as we move forward to make much more safer Rwanda and the region. Thank you.
Great. And just, I think, as probably a brief follow-on on that, there was a question, I think this would be more for Edson, although Mark, you might want to comment on it from some of the other trials. In the Rwanda response, were there any cases that were subsequently experienced among those who were vaccinated? I don't believe you said anything along those lines, but please, could you clarify, confirm?
Yes, but we can't conclude on this. So one of the healthcare workers, just I mentioned, that got infected within the treatment center was actually vaccinated, and but he got the infection just some few days, even less than, I think, within less than 10 days after vaccination. So we can't conclude that there's any kind of how vaccine was effective to prevent infection among people who are vaccinated. So we are working on some of the documentation for this, and we'll be able to share what has been the outcome of vaccination over time.
Great. I think, Mark, there are a couple of questions for you. There's a question from our mailman colleague, Nisha Mishra, about comparison of VSV versus adenovirus backbone in terms of vaccine efficacy and effectiveness. There's another question asking about how long the protection is in animal models for the VSV vaccination. And if you can, and I'll help you to remember my question I'm going to add on top of it, I found myself wondering about the possibility of, if you will, a universal hemorrhagic fever vaccine where you could perhaps either mix the different recombinant viruses into one vaccine or some other approach. I just wondered if there had been thoughts about that. So I've given you 3 questions.
Thanks. Thanks, Nishay, for your questions. With respect to the one about how long protection lasts in the non-human primate models, we only know out for a year. We know that there's still 100% protection at a year out, but that's the latest that the animals has been sort of held before the delayed challenge. So we don't yet know how long protection lasts for in that model. In people, we know from the Ebola Zaire, the Ervebo experience, that immune responses are actually quite durable, and people who are vaccinated essentially 10 years ago have still meaningful levels of antibody responses. With respect to the question about have the vectors systems been directly compared in non-human primates? There have been parallel studies done. There haven't been directly within one study done, but there have been studies, at least in the Ebola Zaire context, comparing Ervebo, the VSV recombinant, with the Janssen vaccine, which is an adenovirus 26 boosted by MVA vector, a prime boost regimen. And there were also studies done comparing the chimp adeno Ebola Zaire vaccine that GSK developed against Ervebo in the earlier Ebola Zaire outbreak. So there are data comparing that. It's hard to know how to truly compare that data because the only vector system and the only target for which there's a licensed viral hemorrhagic fever vaccine is Ebola Zaire. So we can make comparisons to that, but for other vector systems where the action may not be the same, it's not possible to make those comparisons. And Jessica, with respect to your question about having kind of multivalent viral hemorrhagic fever vaccines, I previously had been a bit skeptical about the value of that approach, but when I see what's going on in Rwanda and also in Uganda and elsewhere in recent outbreaks, a number of the people who were impacted, and unfortunately many who died, are healthcare workers. I think that there is value in having broad protection against viral hemorrhagic fever vaccines. And the good news is that you can combine Ebola Zaire, Marburg, and Ebola Sudan, a sort of 3 vaccine combination in non-human primates, and you get 100% protection against each of those individual pathogens when the animals are challenged. So it suggests that a similar approach would work in humans, and we're working to develop vaccines that have that profile.
Yeah, that sounds really great to hear that's underway. Let's see, I think going back to Dr. Edson, there was a question, and some of this has been touched on, but maybe just to expand a little bit more about lessons learned to better protect frontline healthcare workers right at the very beginning of the outbreak. I mean, I can imagine if they had been vaccinated with a multivalent vaccine, that would be a great step, but I understand, that's still in the future. Dr. Edson, what are your thoughts on lessons learned at this point for healthcare workers in particular?
Yes. Yeah, and I think it's a very relevant question since most of the people who get affected or infected frontliners, and don't want those kind of workforce to be affected, since actually it affects the entire health system. So with lesson learned, one of the ICUs were affected, the 2 ICUs, the main ICUs within the country, and we almost had we almost led to kind of close out of those ICU with the healthcare workers being affected and also the ICU themselves. So it was a kind of quite challenge for us. And as we move forward, we have put that kind of triage for everyone. It's with a kind of AI tool that has been put in place that could allow us to at least understand if someone who has any of the symptoms plus working the mining site, at least that would be kind of a high risk person, need further assessment and testing. So that's one approach. The second, we have identified those high risk areas, and since we understand we can have any future outbreak that will be emerging from that areas. So we have put kind of a fever clinics within those settings, at least to make sure that everyone before consulting any of the other health facilities should be going to those kind of clinics and get tested. So those are kind of approach, but we move forward. We're also building the capacity in terms of IPC. And one of the investment that I would like to share with you is our Center of Excellence for Isolation and Management of High Consequence Disease. It's to be launched by December. It's an investment that was supported by Robert Koch Institute, and where we'll be kind of having a cohort structured kind of training for continuous capacity building in terms of IPC. I think with this kind of investment will at least allow us to be able to get much more prepared for any future epidemics and pandemics.
Okay, great. We have a question about the mines where the fruit bats were found. What did the government do with those mines, given the colonies of fruit bats near Kigali?
Yes. So we mapped out all the mining sites that actually the caves that have those fruit bats, and we had provide first of all to enhance our surveillance within these kind of settings. Second, we had to establish those fever clinics, and third, we also had put kind of a buffer area to avoid any interface between the bats and also the human activities. So initially, you could find people working under the places where there's bat roosting, but now we have closed all those areas just to allow bats to still remain there. We don't want to interfere with the ecosystem, but again, we also need to make sure that there's that kind of buffer between human activities and the fruits bats that actually roosting in those areas.
Great. I want to just see if
I have a question, Jessica.
yeah, please go ahead.
Hi, thank you very much, Edson and Mark, for superb presentations and very timely. I do wonder whether maybe a question for both of you is, it seems to me like when you look at that map with the different outbreaks, hemorrhagic fevers across the continent, it's kind of the question that comes to my mind is the need for kind of regional or cross-country collaborations in terms of both early detection, responses, as well as also in terms of sharing of vaccine stocks and so on. So I'm curious from both of your perspectives, do you see that happening, and what more needs to happen to enable the whole region to be able to respond appropriately and effectively to these outbreaks?
Edson, you want to go first? Okay, well, I mean, I guess I can start. I completely agree with you, Wafa. I think that kind of coordination is essential. You know, the Africa CDC has a major focus on viral hemorrhagic fevers, and I think they're working hard to put in place that kind of mechanism. My Eds can comment on what it looks like from the Rwandan perspective. My own sense is I think we have a lot more work to do, and I also worry that recent impacts on global health of U.S. government positions is going to make any sort of more aspirational goals like that even more difficult to achieve. But I know there's a lot of momentum on the African continent to do that. What I think is missing is really kind of a true like strategic approach that looks at how do we make sure we have the best prevention, early detection, rapid response capabilities, but also in the vaccine and therapeutics development, how do we know what the right products are to deploy in those contexts, and how do we enable the development and predictable supply and distribution of those? How do we make sure that there are a stockpile solution available so that the products can be rapidly deployed wherever they're needed? We have a lot of work to do in each of those areas, Wafa.
Absolutely, Mark. I think you have a lot to be done here. If you look at we're doing at least some extent even better in terms of response. Whenever there's an outbreak, we do come together. But when it's in terms of preparedness, that's where we lag behind. For example, looking at the ecosystem, the ecology work that could be done collaboratively with colleagues from Uganda, Tanzania, Burundi, and DRC, at least to understand dynamics in terms of transmission and also the risk of spillovers. That could at least, at some extent, prevent future outbreaks. But we're kind of going the same cycle of response, and we're always called upon to jointly be together when responding, but there's no work really related to how can we come together in terms of preparedness. I think that's where WHO, Africa CDC could also jump in and make sure that we even get much more prepared rather than just responding to outbreak. Yep, I think that's my response. And some of the efforts, I think, with the colleagues from CEPI, we are also working on kind of strategic document for Rwanda for the establishing all countermeasures from diagnostics, development, vaccine, but also most important, the antivirus. So I think it should be one kind of guiding document for us as we move forward. Thank you.
Thank you.
Okay. Well, thank you both very much for just outstanding presentations, and I think we had a very vibrant Q&A session. So this was really wonderful. So as we wrap up here, I will just remind everybody that the webinar recording and the slides are going to be posted probably by the end of today, if not tomorrow, at the ICAP website. And so we will make announcements about future webinars as well. So thank you again for attending and for your attention. And thank you again, Mark and Edson, for such wonderful presentations. Take care, everybody.
Bye.
Bye.
Thank you. Bye.